Hello, students. Welcome to Chapter 2 of Lifespan Development. Let's cover a lot about genetics and epigenetics today. We'll start with a couple of the basic principles that you should understand before we get into some of the more detailed things having to do with lifespan development. You should probably already be a little bit aware of natural selection and adaptive behavior. This is the idea that any individuals in a species that are genetically mutated a little bit so that they're slightly more adaptable in their environment are a little bit more likely to survive and reproduce. So those adaptations come from slight genetic mutations. So as you can see in this picture here, you've got the bird with just slightly different beak sizes. If that's just a little bit better at getting the nuts, it's a little bit more likely to pass on to the next generation. When we get enough kind of similar mutations and we get enough of the offspring with that mutation, it becomes more solid in that population. So this is a pretty long process, the evolutionary process of changing genetics over time. So with this, the focus of evolutionary psychology is an idea of looking at how that kind of adaptation, and mostly where it has to do with like reproduction, natural selection, what that might have to do with human behavior. So, as an example, humans were focused on really specific challenges in our evolutionary past, things that drove the evolution of our brains. So, we did things together as groups like hunting, finding shelter. We formed groups together to be able to survive better, to raise our young. So, humans evolved a really very large brain overall. This led to a long period of childhood compared to a lot of other animals. So I just saw some deer this morning, for example, and they basically grow to adulthood. I forget if it's within 1 year or if it's the second year that they can have their own children, but that's a lot faster than us human mammals. So we had to develop around the things that were adaptive. Our better brain for solving problems, making social groups. This was adaptive for ancient humans. And it still holds on today. So a lot of the things that were adaptive in the past might not necessarily be adaptive now. So as an example, our ancestors, our human ancestors, lived in fairly food scarce environments. And so we made an adaptation to really like fatty foods and high calorie foods. So when we got that, it made us want to eat more as humans. So now we have a few more high calorie foods a lot more easily available. And so we might eat more of it than we probably should because it was evolutionarily adaptive. We get a chunk of meat or some really fatty food. We would eat more of it in our evolutionary past, make it more likely that we would survive, pass on our genetics, that kind of thing. But now it means that when I'm making my kids' lunch and I see the bag of M&Ms up there hiding away from the kids where they can't see it, but I'm tall enough to see it, it makes me want to grab those M&Ms and eat those M&Ms, even though I don't need those. So I'm craving those high calorie foods. So thanks, evolution. Also, our stress response. In our ancient past, most of our stressors were more immediate. So things like a saber-toothed tiger coming out and getting us, or so having to run away from that or being eaten. So our stress responses evolved to kind of flight or fight responses to be pretty quick overall. So we either took care of the challenge or we died, essentially. So you have a challenge, you fix it, and then you calm back down. So in today's world, we have other stresses. You can see a few of them on the slide here. Money stresses, education, maybe you have debt, maybe you know you have a midterm coming up, things like this. These are kind of constant stressors out there. So our stress system has us amped up to fight this challenge of the test coming up, and we're not able to calm down as well. So that evolutionary response might be some of the reason why we have so much more stress and it's affecting us in so many negative ways. So with that being said, while I'm talking about these things that might be from evolution and be affecting us in our modern day, I do want to make it clear, and your book tries to make this really clear. While evolution has created our physical structures, like it's given us bigger prefrontal cortex in our brain, it's given us some propensities in our biology, evolution alone does not dictate behavior. So while we have these capacities, while we might have more stress towards, say, a test coming up, it doesn't dictate how we deal with that stress. We can learn coping skills and tools so we can regulate our emotions, things like that. Evolution doesn't dictate all of our behavior across our whole life. And you'll see that as we go through class today and through this chapter about how much impact environment and yourself have on your genes. So let's talk about the actual kind of molecular structure of these genes. So chromosomes are an important piece of this. They are in every human cell. So chromosome is basically wound up DNA. So the DNA is wound up into these XY chromosomes, right? So what is DNA? DNA is a really complex molecule. It is long, it is in a double helix structure. It's all twisted up into a chromosome. And that structure of the molecule, the order of the molecules in the DNA, contains the genetic info. So that's what a gene is. A gene is that unit of hereditary information. It's a little bit that tells bodies what to do. So I'm not sure why this is cutting off now, but what this says, maybe I could just exit out of here and it should come up. Sorry about that. So protein is the building block of cells. You can just make all of this text a little smaller, and that way it will all fit. Sorry to do this right in the middle of a lecture. But I'll be using this today as well. So we've got chromosomes. We've got the chromosome. I'll show this picture here. So I've got inside a cell, we've got a nucleus. Inside that nucleus, you've got chromosomes floating around. That chromosome is a really tightly packed bunch of DNA. So here we are unwinding it, and you can see that double helix structure of the DNA. The order of these molecules connected with each other makes up the gene. It's the segment of hereditary information. Your body essentially reads that, and proteins can read that and then do something in your body, whether it is ask for more energy, whether it's grow more, whether it's make your eyes a certain color. The proteins are the building blocks of cells. They come and read that information and then go do something in the body. So here we've got a cell, the nucleus with the chromosomes. Zooming in on the chromosome, it is made of this long strand of DNA. And so then we'll zoom even a little bit more. So here's the DNA. You've got adenine, cytosine, thymine, and guanine. So ATCG is what we say shortening it. And so these are all paired up. And so a protein will know what to do based on what order these are. And so there's a ton of genes on our DNA. Again, all of the DNA in all of our cells has all of the blueprint, has all of the genetic code for what to do. That being said, in your eyes, the genes that are active are only certain ones. Your hair, only certain ones. In your muscles, only certain genes, right, are read by proteins, then do things in the cells, so say muscle cells, and that all together makes up our bodies. So the best way to think about this is that the gene is basically a code. The proteins are made up from and can read that code, do things in the cells. Overall, the cells make up our bodies. So humans have 20,000 protein-producing genes. We have other genes too that don't actually produce proteins and don't actually give instructions to bodies. So if you look at the Human Genome Project, you can find some cool things that are in humans' genes that aren't actually active. So like scales, for example. So the genetic code for scales, like some other animals have, humans have that too, but it's just always turned off, or almost always. There might be some cases where it's like partially turned on, but you can find more about that. So genes work with other genes and other non-genetic environmental factors to direct the creation of proteins. So these proteins are doing things in cells. They're telling the cell to grow, to divide, to take in more energy, to do a whole bunch of different things to affect your body overall. So with this, genes are collaborating with each other. It's not just one single gene. It's usually not one single gene making all the decisions. Eye color, while we usually think of it as like very simple, there's one or two genes. There's quite a few genes making your eye color. And that's how they work. A lot of genes making a lot of proteins together, do a lot of things in cells. So any kind of phenotype that we see is dependent on a lot of genes working together to make something happen. So a good example of the environment affecting genes as well is light will affect the hormones in your blood. You've probably heard of melatonin, your sleep-wake cycle. So light will affect those hormones in your blood, whatever is pumping more through it. Maybe it's melatonin, maybe it's not, maybe it's cortisol. And those hormones through the blood get into the cells. And with that, those hormones can then turn certain genes on and off. So make things happen more, make things happen less. So another example of things that in the environment that can affect the expression of genes. So to be clear here, what is happening is this genetic code doesn't change. The environment isn't changing this genetic code. The order of these ATCG, that's totally the same. What is changing is that it might read it differently based and do something different based on the other non-genetic factors. So the specific hormone. They might have a different instruction because there's also this hormone. Some other examples are stress, exercise, nutrition, radiation, temperature, sleep. Some of these are the biggest ones that we've studied in humans and other animals, and these can all affect how genes express. So again, genes are working together. If they're expressing slightly differently, they can do different things in our cells and our bodies overall. So things like stress, exercise, nutrition. Usually the studies as of now are looking at epigenetics of how these things lead to bad things in humans. Oftentimes science kind of starts that way. We see a problem, we say, oh, can this explain some of these problems that we see? And so that's what a lot of research does, but we also see some epigenetic factors that make genes do good things in our bodies too. So having proper nutrition versus not good nutrition, our genes are happier with that and they do a better job with things. All right, so here is a more complicated diagram. If you are a little more into biology, if you've taken some more bio classes, this might make more sense to you. So this is a little more specific of how we get from the DNA to the proteins to do gene expression for the traits that we see and for cells to function. For the purposes of this class, essentially all you need to know is that proteins are complex molecules. You can think of them as reading the code off of the DNA and then doing something in a cell. If you can remember that, you're good to go. And we'll talk about some epigenetics and how these things might lead to that DNA being read in a different way as well in just a moment. So with this, behavioral genetics, in especially lifespan development psychology, mainly uses twin studies and adoption studies. And if you stop and think about it for a few minutes, I bet you can think of why that might be. When you're comparing twins, these are people with identical or really very nearly identical genetic traits. So you can help determine if the kind of behavior between these twins is dependent on the environment or more dependent on the genes. So when looking at adoption studies, we can look at the environment from the birth parents versus the environment from the adoptive parents and get a sense of what is the genes from the birth parents versus what is the environment. You can also put these together. If you have twins that are adopted by separate families, behavioral geneticists love to find that kind of situation because then they can get a lot more data about these kinds of things. Is it the gene or is it the environment? So with this, as we are looking at these genes and the environment, we're not doing experiments here. As we've talked about, experiments are the only way, one of the only ways to determine cause. You can get cause from like longitudinal studies as well, but you cannot get cause from correlational studies alone. So when we're talking about these environment and genetic correlations, that means there's a relationship there. It doesn't mean causation. So what we see is that in our life, we have a lot of environment and genetic correlations. There's relationships between them. And we have a couple names for a few of these. One is the passive genotype environment correlation, another is the evocative, and another is the active genotype environment correlation. So let's talk about those all with a couple examples. So for passive genotype environment correlation, what we're talking about here is that the environment is already supportive of the child's genetic traits. So we might say, what if music? Let's take this example at the bottom. Let's pretend music is entirely genetic. It's not, but just as an example, let's pretend ability to be pitch perfect or have good rhythm. Let's say that's totally genetic. If the parents have those genes, they might already have done things like music. They might already have things in the home to encourage kids to play more music. So the kids here, their organism is passive in that environment, but the environment is already supporting their genetic predispositions. So if you are with your genetic parents, you already might have an environment that's passive to you, passively encouraging your genetic traits. So you have that trait for music, but now you're also practicing. So you're getting the genetics and you're getting the environment to make you better at music. Let's pretend, well, that's not really the picture here. Let's pretend that reading. Let's pretend reading is reading speed. Let's say how fast you read is 100% genetic. It's not. Again, I'm just using this as an example. But if your parents have that gene for fast reading, more likely they will have more books. They will have read more books. They'll take more trips to the library. And so you, as an organism child, you will passively be in an environment that's already supporting your genetic traits. So that's what passive genotype environment correlations is. It's passive for the offspring. It's passive for that one organism. So their environment that they're in is passively supporting their genes. Evocative is a little bit different. In this case, the organism or the child or you as a person, you are actually changing things in your environment based on your genetic traits. And this is an intentional change. This is you just happen to change things. So let's pretend that being friendly versus being rude to friends is totally genetic. Again, it's just an example. This is not really fully genetic, right? But let's pretend it is. So let's say one child has a gene to be really friendly and sociable. Another child has a gene to be really mean to other people. Which one do you think will have more friends? Obviously, the one that is more friendly towards other people. So they have the gene to be friendly. They also are eliciting an environment. They are being friendly. They are getting more friends. So they are evoking, they are creating in the situation an environment that supports their genetic traits versus the one who has the genes to be mean. Well, they're probably not going to get very many friends. They're not going to have as much practice with being friendly. So they are changing the environment in a way that supports their genetic traits. So that is evocative. So the last one is similar to evocative, but it's different in one key way. Active genotype environment correlations is also called niche picking. Basically, it's when a child seeks out an environment that supports their genetic traits. So this is a more active choice. Versus in the first one, you're just good with friends, so you tend to have more friends. And that's genetic. And in that example, that's genetic. Here, let's pretend that, say, gymnastics ability, being able to have kinesthetic awareness, being active and jumping around, doing things like parkour. Let's pretend that's entirely genetic. If you have that genetic trait, well, you might want to pick going to a park or a playground or a big jumping area like this in this picture. You might literally pick that because it seems cool. It's something you can do and you like to do because you have those genetic traits. So the differences here, we have passive, where you're just in an environment that supports your genetic traits. Evocative, your behavior evokes or changes the environment in a way that supports your genetic traits. And then active genotype environment correlations, you are literally picking situations or picking environments that are supportive of your genetic traits. So it's an active process. So that's some ways that sometimes make it hard. So if we go back really quick to these twin and adoption studies idea, this makes it kind of difficult to see, well, how much is genes, how much is behavior and environment? Because we have these correlations, there's a relationship. If a twin is in an environment with parents who also have those genetic traits, maybe it's the environment too. So it makes it hard to determine absolutely what percent, but when we get bigger sample size, it makes it a lot more clear. Like more and more people in the kind of study, we can get a lot clearer answers, and more diverse people in the study as well helps us make a lot better decisions of how much is genes, how much is environment. So another thing to cloudy up this is called epigenetics. This is a really neat way that your environment can literally change the expression of your genetic traits. So this is the bidirectional interaction between your heredity, and my face is probably covering up some of that, but that says heredity and environment. Environment can affect heredity, and heredity can affect environment. So as an example, a prenatal or baby inherits genes. In prenatal development, they might have toxi or healthy nutrition, and that can affect how the genes then express. Some might activate, some genes might not activate based on the environment that it is. So I want to be clear here for epigenetics. This is about changing the molecular structure of the DNA strand, but it's not changing the lines of code, right? So the ATCG, it's always in the same order. You clone that cell, it's the same. But there are other aspects of how this protein comes and reads the DNA that might change how it reads it. So then it does something a little different than you might have expected otherwise without this environmental impact. So this is changing the molecular structure, but it's not changing the line of code. The password, the genome is all in the same order. So we've got a video in a second that will explain this a lot better than I'm doing right now. But hopefully you can see this is another example of a bidirectional interaction. An individual's genetics, so here on the diagram from Lerner's Developmental Contextual Model, we've got biology right there. That biology can be affected by the school network, by the social network, by environment, and that it in turn can affect how the child interacts. So a couple examples from a textbook. This might be from a previous textbook I used in Lifespan. I can't remember. It might be yours. But an example is this gene, the 5-HTT-LPR gene. There's a couple different things that it can do. So if you have a stressful life, so extra stress in your life, more cortisol in your bloodstream, in the hormones, it makes this gene activate, and it makes you more likely to suffer from depression. So again, a lot of the studies we do on this are looking at negative outcomes. Well, could epigenetics explain this negative outcome? And the other interesting thing, that same gene, a longer version of it, is protective against parent loss. If it's shorter, it's not so good. And that can change based on parent loss or based on environment, based on stress. So your environment can affect the ways these genes express. So let's watch some video examples to get a better sense of what we mean by these genes are expressing differently. So remember, the code is the same, but the genes are being expressed differently based on your environment. All right.
So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, clone probably has a tattoo on its face. And if clone had stayed in the circus, clone probably, professionally rides a unicycle over a tightrope, so clone is probably in pretty good shape. But then again, clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So at 50, you and clone would probably look pretty different from one another. But it turns out you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that of the person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference. And if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely potentially changing the message of that paragraph.
All right, so going back to these images, of the DNA. Just to be clear, what we're saying here, let's get this one and I'll scroll back quite a few. So what we're saying here is that these letters, or these letters in the paragraph, are the letters of the genetic code, are the letters on the DNA, are the ATCG. That order doesn't change. So you clone a person, you have the twins, and twins can have some slight variation, but that's beyond the point. You have a clone, the same exact genetic code. So with that, those letters stay in the same order. ATCG, whatever order it is always the same, but there are things that can happen to that DNA strand. Remember that DNA strand is wrapped together into a chromosome. It can be wrapped more tightly, it can be wrapped more loosely, it can be kind of stretched in different ways. So in some cases, it might be read differently. The protein comes, tries to read 3 letters of the code, but they're actually getting 4 in that same spot. That's an example of this.
Study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob.
So with that, going back to this image, there are literally, some of this is a basic, just biological, physical process that the DNA is too tightly wound or too loosely wound and is read differently by the protein. So this protein is expecting to read a certain code. It comes to read that code, and there's more or fewer letters there, molecules, the ATCG. And so now it reads that instruction differently than it might have otherwise.
The switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern, and that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life, but these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary, and it can change over time, especially when your body is going through a lot of changes, like say during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're gonna have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're gonna have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very...
So with that, actually, Hank Green here just recently overcame cancer, went through cancer. I forget the terminology. Came out on the other side, clear of cancer. So that is great to hear. But going back to this, we have in each of these cells, we have this nucleus, We have chromosomes in each of the cells, and each of those chromosomes is built of the same DNA, all the same code in every single one of your cells. So these methyl groups are blocking off a majority of a lot of the DNA so that your eyeball cell or your tongue cell or a muscle cell knows to be that kind of cell. So again, like I said, environmental factors can make those methyl groups go to a different spot, but usually what we see is these histones, is the winding or unwinding of DNA to be more or less tight make a difference in when this protein comes in. So this big globular thing, it's again just complex molecules coming and reading the DNA and then going to do something in the cell. You might read it just a little bit different and go do something a little bit different in the cell. When we have that enough, we can see some epigenetic changes. So the next point that video is going to get at, feel free to watch the whole thing. It is really great. The next point is that we used to think that these epigenetic changes only applied to us and that they did not get passed down. But something that we're seeing is that some epigenetic information of how tightly to wind that, where to put the methyl groups, some of that can get passed down to offspring. So some of the environmental things that I am doing today, well, I'm not going to have any more kids, but some of the things I did 20 years ago were affecting my histone pattern, my methyl groups, so that my offspring might have some of those epigenetic tags. So it's a really cool area of science. We're getting better and better of measuring the things that have to do with epigenetics, but I also want to give a really concrete example of epigenetics in a couple animals to hopefully make this more clear to you as well. So let's jump to another video. So this whole video is pretty funny. It's all about looking at epigenetics in the sense of Eevee. So if you play Pokemon, I play Pokemon Go quite a lot. Eevee is one of my favorite characters. It can evolve into a lot of different things. So it's kind of teaching you what epigenetics is with Eevee as an example. But we're going to look at a couple of the actual examples that we see in nature.
So evolution alone isn't explaining how Eevee's physical changes are happening. But believe it or not, there is, in fact, a scientific explanation for Eevee, which is good because I like Eevee and we already wreck too many nice video game characters on this show. It's called epigenetics. Epigenetics is something that you don't usually get into unless you're majoring in this stuff in college. So all you younger viewers, get ready to blow your teachers' minds when you drop this knowledge bomb in the middle of your lesson about Punnett squares. Those are so like 4 years ago on this show. So what is epigenetics? At its core, it's essentially the phenomena by which animals are born with one set of genes, but potentially have parts of their DNA that are locked at birth and can later be unlocked by certain things happening in the environment. If that sounds weird, think of it this way. You have a ton of DNA in your body, 2.9 billion base pairs, but
so with that, yep, you've got that many base pairs of DNA. Remember, we only have about 20,000 protein producing genes. So about 20,000 specific instructions for what our DNA should do across our whole body, which is still a lot, but just to put things in context there.
not all that DNA is working all at the same time. Without getting into the nitty-gritty of genetics, your cells have specialized proteins that turn off and on different genes throughout the course of your lifetime.
So here, these are the methyl groups. I don't remember if this specifically says it in the video, but that's the on-off switch. So do not express this gene, do express this gene, that's the methyl groups. And then the histones, again, were the one that was like a knob. It's literally twisting the DNA tighter or looser.
When you're a kid, you have a ton of genes turned on to do things like grow your bones, build your brain cells, and create the hormones that make you fall in love with literally every creature that you pass in the school hallway. Later in your life, though, some of those genes get locked back up. When that happens, maybe your metabolism starts slowing down. You stop producing color for your hair, and it turns gray. As you get older, you have all the same genes that you started with on the day you were born, but how you look is depending on which ones are turned on and off. Your epigenetics.
So I can blame epigenetics, at least in part, for my hair thinning and turning gray. But honestly, my hair's been turning gray since I was like 13. So that one's fine.
Right now, my baby Oliver has blonde hair, but Steph and I have brown hair. So should I be worried about Steph's relationship with the milkman? Is Steph cheating on me? Of course not. At least I don't think so. Steph! Steph, are you cheating on me? Oh, yeah, sure. That's what you would say. No, what's really going on here is that right now, most of the genes in Oliver's system that produce melanin, which turns your hair brown, are turned off. Like, way off. Just like I had when I was his age. But when he gets older, those genes get activated. And his hair will turn brown just like ours. His physical traits will change as his genes turn on and off.
All right, so that's a really good concrete couple of examples of human genes. Let's see some really interesting examples of the environment dramatically affecting the genetic expression of a couple different animals, which actually I think the first one, yep, is actually right after this portion. So it's about bees and how they change based on just one environmental factor. It's pretty dramatic.
Now, with animals, what epigenetic researchers have discovered is that some animals will be born with one set of genes, but potentially have parts of their DNA that can be unlocked by certain triggers in their environment. A great example of this are honeybees. Yeah, I know. I just dedicated an entire episode to bee talk as it relates to Peachette. Maybe I'm just going through bee movie meme withdrawals. I don't know. Bees are fascinating creatures, and in this case, they are the perfect EV example. So, like I covered back in that episode, female bee larvae can grow into either a queen bee or a worker bee. What makes the difference here is the food that they're given at birth. Bees given royal jelly become a queen, while everyone else becomes a worker. So in this case, royal jelly is basically the evolutionary stone for bees, determining what form the larval bee evolves into. It even changes the bee physically, where in their adult stage, queen bees and worker bees look way different.
So this is a pretty dramatic example of one single environmental factor in that nutrition that it's getting will change the way that its genes are read. So all of the bees in a hive are siblings, right? They're very, very similar genetically. But even if you took two twins or cloned a bee and made two larvas, if you give, so you've got two clone larvas, you give one the royal jelly, you give one just the normal food that they give other bees, honey, sugar, whatever it is, the one that gets the royal jelly, its genes will be expressed differently. So it will be bigger, its behavior will be different. So queen bees act different than the worker bees. So it is dramatic that this one environmental factor will change how these genes are expressed. And again, this is from the methyl groups, this is from the histones, reading proteins reading the DNA in a slightly different way to do something different in a body. And here we see both behavioral and physical changes. There's another example later on here that I wanted to show. It does a good job of showing the histone patterns. Oh, it actually is right after. I thought it was further on in the video. So this is a lot better job of showing the histones, or that knob, having subtle differences in genetic expression based on environment.
But in their larval stages, they're practically identical. It's exactly like two different evolutions that wind up with two different appearances despite starting with the same looking EVs. The queen and worker bees didn't start off as genetically different, but differences in their environment, the royal jelly, or lack thereof, led to wildly different gene expressions. And bees aren't the only place in nature where this happens. Another EV-like example happens in the agouti mouse, which has a gene which changes the fur of its coat. These two mice are genetically very similar.
So when he says very similar, in the actual study, these are siblings. Like these mice are siblings of one another. So you could do this by cloning mice and getting the same result. But in these studies, it costs less money to use siblings. And you still get a lot of the same information. But the same thing would happen if you had total clones. If you had a whole family of 20 mice clones, that they were exactly genetically identical, you would still see this phenomenon happen.
At birth, but due to differences in nutrition, they have very different gene expressions, with one having a gene expression that causes it to become obese and have lighter fur. Same DNA sequence, different phenotypes, all due to environmental factors. Very different gene expressions, with one having a gene expression.
So here we go. So you can see the mice's coats, how dramatically different they are. So this, if you looked at this one compared to this one, you might think, oh, those are very different mice. But you can see the kind of range as we go across this. And that is based on that histone pattern, how tightly or loosely that the DNA is wound. I read this study years ago. I can't remember. I think it was the tighter it was wound, the darker the mouse versus the looser it was wound, the lighter the mouse. So essentially what the proteins that are responsible for making the color and size are doing, they're reading those different parts of the DNA and then collaborating together to create the darker mouse versus the lighter mouse. So because the DNA is reading it slightly differently, but remember the code is the same, but that protein might be getting 4 versus 3 letters that it's expecting, and it leads to this dramatic difference. And so this is still a new science. Again, with a mouse, we can do a more controlled experiment. We can clone them even if we wanted to and test out different environments. In humans, it's not really ethical to do that. So we have to do things like twin and adoption studies. We can look at the human genome and look at changes over generations, looking at people's DNA with, say, blood samples or skin samples and things like that, which is really, really neat. But these kind of epigenetics are happening all the time. We have the environment affecting the way our genes are expressed. So this is probably beyond what you had ever considered of how genes might affect your body. And hopefully you can see as we go through this class overall and looking at development, how not only does it change how we necessarily think about things in psychology, right? Lifespan development, your parent interacting you with this in a certain way, maybe you have a certain stressed versus not stressed childhood growing up. That's not just affecting your brain's response to this. In some cases, it's affecting your genes expression, your genetic response to things as well. And some of those things can be passed down later on. So with that, you can add this to your idea of this developmental cascade model. So the genes are part of this. Maybe you have a range of possible genetic expressions, kind of like the mouse. You could have very dark mouse over here, very light mouse over here, depending on your input from the environment might depend on where that little water molecule falls along that genetic expression. So development is complex. We have multiple genes affecting any given trait that we see, but we also see that complex behaviors are influenced by genes, but it's not just one gene affecting a behavior. Even though my earlier examples, I'm like, if you have the genetics for being a nice friend, it was just a simple example for that, that thing. But really, complex behaviors are dependent on a lot of genes. So those genetics might push us one way or another, like nudge us in the developmental cascade, but it also requires environment, it requires our own decision making. So it's a very complex process. It's not just genes in this image. So it's the genetics, it's our experience of the thing, it's our interpretation of the thing, it's the supports or lack of supports that we have, the other risk factors or protective factors. So in the end, like so many things we're talking about, genetics are affected both by the environment and they are influenced, or it's nature and nurture. Development is genes and environment talking with each other, interacting with each other. But we as humans, and this is a point brought up in your book, and I basically just copied and pasted the sentence from it, but we as humans, we can author a unique developmental path for ourselves by changing our environment. There are things that we can change about what we do to change how our genes are expressed, to change how we feel about things, change how we are experiencing things. Some dramatic ones might be therapy or a new diet that's healthier. These are ways that we can change our environment in a way that can be beneficial to us.
The rest of the chapter is really focused on the prenatal development as well as post-birth period. So let's talk first about a couple of the phases of prenatal development. We've got the germinal period, the embryonic period, and the fetal period. So with this, the germinal period is just the first 2 weeks after conception. So oftentimes at this time, a person won't even know that they're pregnant. So here we have the fertilization of an egg. Now it's called a zygote at this point after it is fertilized. We see cell division happening, and we see the attachment of that multicellular organism onto the uterine wall. So this is the process that it takes. So you can pause and look at this in more detail. I believe this diagram is also in your book, but that's the germinal period. So first 2 weeks, you get that zygote into the uterine wall, essentially, after that fertilization. The embryonic period is a little bit longer. So it's that 2 week spot to 8 weeks after conception.
So now what we call it is an embryo. There it is implanted into the uterine wall, and it is really going from this multicellular organism into this embryo that looks a lot like, we can see some features that look like a human. We also see features that a lot of mammals actually look very, very similar at this embryonic period. But a big point of this period, and you can see some more specific details if you pause. I won't actually read all of these things. I believe, again, this diagram is in your book if you want to look at it in your book as well. But a key part that's happening in this embryonic period, remember this is from 2 weeks to 8 weeks, so still very early on in pregnancy, is that organs are starting to develop. And so this is one of the periods where they are most susceptible to environmental influences called teratogens. And we'll talk about teratogens in a lot more detail in just a minute. But basically, things like alcohol can really negatively impact the development of those organs. And so this is hard because this is a time when a lot of people who are pregnant aren't necessarily going to know yet that they're pregnant. Oftentimes, people figure out sometime during the embryonic period after they've, say, missed a period, and then they wait a little bit and they test. If you have some people who are planning and trying to get pregnant, they might know earlier on. Right? They might know pretty early on, even before the embryonic period. But for a lot of people, it's towards the end of the embryonic period or even into the fetal period when they realize that they are actually pregnant. So, embryonic period, key part here is that especially a lot of the parts of the brain, a lot of the organs are starting to develop. It's still a time when those organs are very susceptible to outside problems. So fetal period starts at that point at about that 2 months, and it lasts the remainder of the pregnancy. So again, you can see some more details here. There's a lot of things that happen during the fetal period. So we've got the second trimester, the middle 3 months listed here. We've got the third trimester here. Again, feel free to pause and read these in a little more detail. The thing I want to focus on is actually the development of the brain during the prenatal period. So here are some diagrams of brains throughout the prenatal period. So the basic structure of the brain, so like the different parts of the brain, are mostly in place after the second trimester. And at week 5, what we see is a ton of new neurons being developed. So in humans, it is kind of interesting. We are finding now that some neurons are being produced. It's called neurogenesis in our brains throughout our lifespan, but only in a few key areas. Most of the neurons that we have are happening in between conception and age 2. So, and really most of it is happening prenatally. Like we do still have neurogenesis happening after birth and up to about age 2, but it slows down dramatically. And you can kind of see that in these diagrams. Actually, I will get to this bigger slide. So we can see just based on the differences of days, where's my mouse? There we go. We can see the basic structure. You can see it even here at 20 weeks, 6 months. This is about when the child would be born for a full-term birth, right? At 9 months. And there you can see it. The real big differences between here at 9 months and a young child, and I'm not sure how old this young child is, let's say 7, probably. Essentially, a 5-year-old, 7-year-old, their brains are going to look pretty similar. You can see they do have more neurons here, but most of their neurons are already there. At birth, you've got a ton of neurons, a ton of connections already. And there is my previous lecture recording finishing being saved. So here at 9 months, you've got a lot of the neurons that you're going to have your entire life. So especially during the embryonic period where the brain is just starting to develop, it is very, very susceptible to teratogens, to outside influences. But it's also really neat to think about what is the brain doing during that time. Most of what the brain is doing during this first period and into the embryonic period is making new neurons. Essentially, stem cells become neurons. Stem cells become all sorts of cells, but stem cells become neurons, and then those neurons basically migrate to where they will live for your entire life. They'll move to the prefrontal cortex
up here. They'll move to the occipital lobe back here. They'll move to the brain stem. They kind of get produced back here. There's a couple places, but a lot of them get produced back here towards the brain stem. Again, there are a few places that the stem cells divide and then move. Because neurons themselves, the individual neurons, our neurons don't go through the separation. So the neurons that you have, they don't divide to make new neurons. Stem cells will divide, and then those stem cells could become neurons, but it's the stem cells that would divide. The neurons don't actually divide. So that's why you're kind of stuck with the neurons you've got throughout your whole life. But that's also one of the reasons we evolved to have so many developed so early on in life. Again, like I said, by age 2, you basically stopped producing new neurons. We are now able to measure that we do have some neurogenesis in specific areas of our brain, but the majority of your neurogenesis, stem cells becoming new neurons, is happening before this 9-month period, and then there's still a chunk happening up until about age 2. So some cool things about this is you've got tons of new neurons being developed. They migrate to their final locations. And then what we can see in fetal brains is between about week 23 and 25, we can see these neurons starting to reach out to other neurons to make connections. Now, at that point, I mean, you can't ask a fetus, like, do you have any experiences? One, they're not going to understand English. Two, you can't do that anyway because you can't, it's not going to happen. You're not going to be able to ask a fetus what they're experiencing. But probably when they're just starting to make connections, it's the brain just starting to wire itself up. But it is pretty neat that we can see these neurons making connections with other neurons, sending messages back and forth. Is there any kind of conscious experience at that time? Probably not. Almost certainly not. There's not enough wiring up that's been done yet. But at a certain time, there would be, right? At some point, there'll be enough connection altogether that you've got an organism that's experiencing things. So with this, the cool thing is that this connection, this continued sending out dendrites and axon branches to make connections with other neurons, this is happening all the way up to age 2. Depending on which area of the brain, some of these areas are still sending out a lot of connections even up to age 4 and 5, but usually at about age 2, for most areas of the brain stops putting energy into making new connections and starts pruning away ones that aren't being used. So with that, throughout our entire lifespan, we can gain and learn new things, develop new connections, but the brain's primary focus through this time is we've got primary focus of making brain cells, making those neurons, and then about this time, both making neurons and making connections, and then after a young child, pruning connections, and then making new important solid connections throughout our lifespan. So that's kind of the basics of development of the brain, and we'll come back to this as we go through the class and in the next couple chapters, because it is really dependent on a lot of the factors that we experience in our lifespan development. Our brain development affects our adolescence, our puberty, our adulthood, menopause. So our brain development and changes over time affect cognitive things later on in life. We talked about in chapter 1 how our cognitive capacities can remain throughout our life if we're working on it. Essentially what that is neurons making new connections throughout life and keeping those connections strong. So brain is one of the main things I'll come back to a lot of times, and that neurobiology of what's happening. But I just wanted to show this image and get a sense of what's happening to the brain, and especially as we get into teratogens in a couple minutes, why this embryonic period is so susceptible and such a big thing to be worried about, especially because it's oftentimes a time when the pregnant person doesn't know they're pregnant yet. So with that being said, let's talk about the experience of the parents on the side of prenatal development. So infertility is pretty common, more common than I realized until I took like child development classes, but 10 to 15% of couples do experience infertility. So that's just defined as the inability to have a child after 12 months of regular intercourse and no contraception. So essentially you're trying to have a baby for 12 months, you're not able to, you'd be clinically defined as having infertility. And there can be many, many causes of infertility. Sometimes it's a couple together, sometimes it's just one, sometimes it's more complicated or easier to
work with a doctor for, but all of these, not ovulating, having abnormal ova, blocked fallopian tubes, implantation problems, you might have too few sperm, the sperm aren't moving adequately, or the sperm are blocked. So if you have one or two of those, you're essentially going to a fertility specialist to figure out what are the problems you're having, how can we fix it. So if there's no ovulation, there's a pretty simple drug that you can take to have more ovulation. And so with that, that might fix it. That might be the first step in doing that. But it does happen to a lot of people. So with that, there's anything from both behavioral changes, like make sure you're getting enough sleep, you're getting proper nutrition, these kinds of things, but also medicines, to all the way to surgeries, and hormone therapies can also be effective depending on what you're needing. So again, those hormone therapies can be as simple as just taking a pill so that you are ovulating more regularly or you're ovulating more, then all the way up to in vitro fertilization. So we've got the eggs and sperm combined in a laboratory setting. So then if the fertilization is successful, those eggs are then transferred to the uterus. So that's kind of like the most intensive or most invasive method of infertility treatments. But it does happen fairly commonly. So something to be aware of. And that can be hard on a person going through infertility. Oftentimes people don't necessarily talk about their challenges with infertility, but it can really affect people. They can be very sad or distraught about this. So something to be aware of, that it is fairly common. Definitely encourage people to seek treatment, because for a lot of people, they might have very simple fixes, you might say, to their infertility. Some might not be simple fixes. I had a family member that was able to just take a pill. Within a couple months, they were able to have a baby. I had another family member that had to, because of costs, go to another country for in vitro fertilization treatment. I don't remember if they went 2 or 3 times because it was such a challenge for them to get pregnant. But yeah, they went at least 2 times to get different separate in vitro fertilization treatments. And they were able to have a baby. So it can be a big challenge for people. So teratogens, I've alluded to them a couple points, a couple times already. Essentially, a teratogen is anything that can cause a birth defect or negatively alter behavioral and cognitive outcomes for that fetus and for the child after it's born. So there's a couple of key things about teratogens. The dose is important. Usually the greater the dose, the greater the effect. Usually, again, that's negative effects we're looking at. So the greater the dose of that chemical, the more problems you're going to see. Also, genetic susceptibility. It is possible that someone might have genes that are more or less susceptible to a specific kind of teratogen. So is that pregnant person's embryo genotype susceptible or not? Another is the time of exposure. At what time in development, at what time in the embryonic period or the fetal period, what time are they getting those chemicals or that exposure to the chemical? Because depending on when it is, getting that exposure might not matter anymore because of the way the physical development is, versus it might be very critical. So like we said, the embryonic period is one of the biggest times of exposure time. So this chart, again, I believe this is also in your book, so take a look at it. As you can see, this red, this greatest sensitivity to teratogens, is the red lines, and it pretty much all embryonic period. There's some that extend into the fetal period as well, and some of these can also still be affected, especially the brain, to teratogens all throughout the process, but there is the most challenge or risk in the embryonic period. So you can see some diagrams here of the weeks of development. So you've got the heart developing, the eyes developing, the brain developing, you've got limbs developing as well, the eye, you've got the ear, you've got teeth and other organs here at week 6. Week 7, you've got basically some other skull bones, other organs, and then the brain this whole time. The brain is, again, throughout this whole time, it's making tons of new neurons, migrating them to where they go, and then only about right here is it starting to try and make connections with other neurons. So after there's kind of enough neurons around to find connections, it starts to make those connections. So with this, there is a lot that could potentially be impacted by teratogens during this time, a ton of organs that can be impacted. So Let's look at a couple teratogens, and I'll put the slides up for others, but we won't go in detail about all of them. So some that are pretty obvious are things like alcohol. So fetal alcohol syndrome disorders oftentimes cause facial deformities. And so let's scroll back here. That can impact things like your palate and skull development, right? Defective limbs, face, heart even. Oftentimes learning problems and intellectual disabilities because of cognitive changes to the brain and the ways the neurons have now moved and are connecting with each other, or different aspects of that. Oftentimes with fetal alcohol syndrome, we see a shorter life expectancy. And so and you can't see the whole recommendation. Basically, 1 or 2 servings of beer or wine a few days a week can cause problems in this embryonic period. So essentially the recommendation is no use of alcohol. And this can be a problem because remember, at this time in the embryonic period, a person might not know that they're even pregnant up until this 7 or 8 week or even into the fetal period. So that's why some of these teratogens can make changes and cause problems, especially if it's not something planned or not something you're expecting to happen to have a baby. So another teratogen that you might not have considered is caffeine. So there's not, for each of these teratogens, we can't do experiments like controlled experiments. You can't take 100 pregnant people and say, hey, you 50, you drink 3 shots of whiskey a night and we'll see what it does to the fetus. You 50 pregnant people, you drink only water and we'll see how the difference is between you, right? You can't do that study. You cannot do that as an ethical experiment. So a lot of this data is correlational, and so it can be hard to find and track causes over time, even longitudinally. So with that, we don't have a lot of good data on caffeine as a teratogen, but the U.S. Food and Drug Administration recommends sparing or no use of caffeine. That it might be, there's some correlations with problems, but it's not super clear. So another example is nicotine. We often see nicotine more used with adolescent pregnancies, and also an adolescent pregnancy or a person within pregnancy in adolescence is often less likely to know as early on that they are pregnant. And so this has increased risks of SIDS, low birth weight, fetal and neonatal death, ADHD, and smoking for that child. Again, these are correlations. Some of them might be related to one another. Some of them might be causal. But when we look at something like ADHD here, this is a correlation. There's a correlation to nicotine use and ADHD. Does that mean that it causes it? No, right? It could, it's possible, but we don't have the data to tie that cause. It might be that the adolescent parents who are also using nicotine also have a genetic susceptibility and have ADHD themselves, and so that's why we see a greater proportion. So when you see these are correlations, these are risk factors. It doesn't mean necessarily that nicotine will absolutely lead to them or is necessarily the cause of some of these risk factors. So again, the recommendation is no use of nicotine. The recommendation is no use of cocaine as well. So oftentimes pregnant people in poverty are using cocaine, and that being in poverty, well, one of the bigger groups that is using cocaine during pregnancy is people in poverty, and so also they might themselves be malnourished. They might also be using alcohol. So there's a bunch of risk factors that come along with poverty as well. So cocaine definitely impacts brain connection and development of motor functions as this fetus is making those organs. So a really sad thing with some of these, especially like cocaine, for example, is after birth, babies suffer from withdrawal. So as the pregnant person, you might be able to say, wean yourself off. They don't do that with a new baby. So after this baby is born, they will go cold turkey on these drugs. And that is incredibly stressful for any person that has used a drug like this and has gone cold turkey. A very, very stressful time for them. But that is the current medical practice, to just go cold turkey. So that's a ton of stress for a organism who just went through the birth process, is now in a completely different environment, now doesn't have this chemical that the brain thinks it needs. That's a tough time.
So do not use cocaine when pregnant. Marijuana as well is one that's been researched more and more, especially as it's become more legal across the United States. So with marijuana, again, we have some correlations. It's not super clear what this might affect in the development of a fetus, but the recommendation is no use. So with that, if you do use marijuana for sleep or diet or whatever it might be, just like any medicine, you can find alternatives with a doctor. And so, for example, where for my children, my wife had to take a different medicine, like shift over to another medicine when we let the doctor know we were going to try and get pregnant, because that medicine would have been the original medicine, medicine A, would have been a teratogen for any embryonic and fetal development. So we're able to switch to a different medicine with the doctor. It was totally fine. So same thing with like marijuana. If there's something that you're using medically, let doctors know that you're planning on getting pregnant. So then they can let you know, oh, do we see some risk factors for this medicine? And let's find an alternative before you're pregnant. Because sometimes, especially depending on what kind of medicine it is, it might take some time to find one that works well for you. So definitely let them know in advance. So heroin is another example of one that can lead to withdrawal symptoms as well. Again, no use of heroin when pregnant. Opioid use has been increasing in the US as well for pregnant women. And so we've really only started the study here, but some very a lot of really bad problems from opioid use in embryonic and fetal development as well. But these kind of chemicals that a parent is putting into their body are not the only potential problem. There are other parental factors that can be, one, teratogens or just additional risk factors. So not all of these are teratogens. So remember, the definition for teratogen is anything that can cause birth defects or negatively affect cognitive or behavioral outcomes. So some of these do fall under that teratogen. Some of these are just additional risk factors that we'll talk about as well. So parental factors such as syphilis. So some kinds of diseases can affect the development of a fetus or an embryo. And so you can see some risk factors for syphilis specifically can cause blindness. It can be treated pretty easily with penicillin. Genital herpes can be contracted in contact with the birth canal during the birthing process, and it can lead to brain damage for a new baby, this kind of thing. HIV and AIDS can get across and infect a child in 3 different ways. So it can affect across the placenta. If that doesn't happen, it can happen during contact with blood or fluid during delivery, and it can also happen through breastfeeding. So if a doctor knows that a patient is HIV positive, then they will take extra precautions to try and be careful so that the child doesn't necessarily get that infection as well. So you can be infected and symptomatic, infected and asymptomatic, and not infected as well. So those are a couple other parental factors. Some additional ones are maternal diet. So we see that obesity for the parent increases rates of hypertension, diabetes, and respiratory problems for the child later on in life. So again, these risk factors, some of those are genetic, right? We talk about epigenetics. There might be some genetic factors or epigenetic tags that have come from this parent to the offspring to make it more likely that they'll eat more, that they want more fatty foods, right? So it's not necessarily just the diet during pregnancy, but we do see a correlation, a relationship between the diet during pregnancy and these outcomes. So we see stillbirth, preterm birth, what a zygote, an embryo, a fetus needs: more folic acid, more omega-3 fatty acids. Those are really good for those developing neurons. Lots of protein, vitamins, a lot of vitamins, and a lot of minerals like iron that you would need more than you would necessarily need as a adult person. So with that, a lot of times simple prenatal vitamins are all that is needed to add these things to a person's diet. So taking those kind of prenatal vitamins is great. I don't think I have in the slides, but overall, people make the joke about like, you're eating for 2. In reality, it's different at different phases of the development, right? So you've got a zygote that doesn't need like any additional eating. You've got an embryo or a fetus, it might need a little more, but if I remember correctly, it's like 300 extra calories a day. So it's not a ton more calories a day. A bigger thing to focus on is just eating healthy and eating enough for yourself, uh, but then also getting these additional things. So getting that iron, getting those minerals, getting those vitamins, getting protein, getting those omega-3 fatty acids and the folic acid. So it's not actually eating for two. In some cases with these vitamins, it's like uh, compared to what you would need, it's like 10 times what you would need. So you could say for the vitamins and minerals, you might be eating for 10 for the vitamins and minerals. So another risk factor that we see is parental age at the time of giving birth and going through that pregnancy process. So the two times that we see the highest risks are in adolescents and for people who are 35 and older. So for adolescents, oftentimes they have higher infant mortality rate. Oftentimes we see that they have not as good prenatal care during adolescent pregnancies. Oftentimes they're less likely to seek prenatal care. We see higher rates of Down syndrome as well. So with this, for an adolescent parent, they might, one, not realize that they're pregnant for quite a while, so they're not starting prenatal care when they should. They might be worried that now they're pregnant. What in the world did they do about this pregnancy? They might hide it from people, hide it from caregivers, hide it from doctors, and not tell people and not be caring for that embryo or fetus. So that is some of the biggest risk factors for those kinds of pregnancies. When we see after 35 years and older, we do see higher rates. So again, these are correlational. We do see higher rates of Down syndrome, lower birth weights. We see earlier deliveries. So that's what preterm delivery means. And when we see a little bit higher risk at 35 and then quite a bit higher risk after 40. So we see increased miscarriages, early births, so preterm births, stillbirths, and fetal growth problems. So with that being said, if you are, let's say 35 and you let your doctor know that you are planning to have another baby, they'll probably just plan to do more testing to make sure things are going well, to make sure that you're on top of your prenatal care. So you definitely can get pregnant after 35, even after 40. There's just additional risk factors. So you need to do more things to be protective for those pregnancies. Again, for the adolescents, the protective factor is care and concern of the people around, like being on top of helping a person get to prenatal care. If it's a person that isn't able to drive, has never gone to a doctor before, never dealt with insurance, they probably need supports because they've never dealt with those kinds of things, right? So helping them out with those kinds of things through the pregnancy. So there are other paternal risk factors as well. So from the sperm side, so exposure to lead, radiation, pesticides can cause abnormalities in sperm. So these can, in some cases, lead to miscarriage, childhood cancer. They can lead to just infertility in general. The relationship with the mother, so the social relationship with the other parent is a big part of this too. So if both partners are supportive, egalitarian, have a positive attitude, and take care of physical and emotional needs, these are all protective factors. Makes things easier for the person who's pregnant. And so that, while this has been researched more in like men and women, this goes for any gender, any partner of the pregnant person. So having a supportive egalitarian relationship with that pregnant person is going to be helpful through the process. All right, so I mentioned prenatal care. What are a couple things to look for? Well, it can be very different for each pregnant person, right? There might be additional testing if you're over 40. There might be more training on, well, how does insurance work? How do you fill out forms for someone who's, let's say, 14 and pregnant? So, but generally, this involves doctor's visits where they will teach you what to expect throughout the pregnancy. You'll probably look at fetal development. They'll tell you your baby's this size, the size of a kidney bean, or the size of an orange, or things like that. Some of these might be group visits. It depends on the doctor and the care plan you choose. Throughout this whole time with prenatal care, exercise is important. With that, we see things that are using your body, cardio, muscles,
These are important. You don't necessarily have to stop, say, weightlifting, though your doctor, depending on your body, might give you recommendations. So if you're, say, a competitive power lifter, that kind of person might have to cut back on like the amount of lifting they do. Balanced diet is also very important. So for actual birth process, there's a lot of options, and a lot of them have a variety of benefits and risks. One of the big things that we're teaching you in this class is how to find research. So you can find some good, you can go to the medical journals specifically and look at the outcomes, look at the most recent data. Hopefully your doctors or your midwives or your nurses, hopefully they will have looked at the most recent data as well of what are the benefits and risk factors. So you can have medication through delivery. You don't have to. You can have a birth in a home, in a hospital, natural childbirth, prepared childbirth, Lamaze, cesarean delivery, water birth, massage, acupuncture for reducing pain. There's a wide variety of ways that you can go through the birth process. So check out this site, babycenter.com has a lot of good, pretty up-to-date data on it. They actually have a nice app. I remember when we were pregnant with our kids, we had the Baby Center app, and it would give us reminders of like, hey, remember to be focusing on this thing. So that was really nice. So I'm sure they have some stuff like that still. So with that, let's talk about the infant side on after birth. So a low birth weight is anything less than 5 pounds and 8 ounces. Very low birth weight is anything less than 3 pounds 4 ounces. And extremely low is 2 pounds 3 ounces. So anything that is lower than kind of average and expected is a risk factor. And the smaller it is, the bigger the risk factor. And if you consider this, well, that might mean organs have not developed as much as they should yet. It's not as it was a fetus, it wasn't getting as much nutrition as it should have gotten, these kinds of things. And that can lead to maybe that embryo and fetus didn't have enough energy to do all of the brain development it needed to do, or all of the organ development. So the lower and lower the birth weight, the bigger and bigger the challenges, essentially. So preterm, so early, is considered 35 or fewer weeks after conception. And about 10% of babies are born preterm in the United States. In other countries, it's closer to 4 or 5%. A lot of this comes down to the quality of prenatal care. So that is something to be aware of. A lot of countries in a lot of medical outcomes do a better job than the US. So most preterm babies, if you're born early, oftentimes you're born lower weight as well. With that being said, those last couple weeks, the baby is really packing on the pounds. It's really getting a lot bigger and heavier pretty dramatically towards the very end of that fetal period. So if you are too soon before that full term, you're going to be smaller and have a lot of risk factors associated with it. So low birth weight is associated with lower survival rates. It's also correlated with learning disabilities, being neurodivergent, breathing problems. But that being said, there are a lot of treatments for being low birth weight and preterm delivery. And I'll show both short-term and long-term benefits. So NICU is one you've probably seen. So neonatal intensive care unit. So basically putting the baby in a box, making sure it's getting the fluids and the nutrients that it needs. Another one, if the baby isn't as high risk, doing more kangaroo care. So that's an example here. You can have the parent themselves or even nurses do this as well. Having the baby close to a person, a mother, with the nutrients getting to the baby as well. So it's kind of a mix of having the baby out as well as NICU. Massage therapy. There's a variety of ways to help support a low birth weight and a preterm infant to getting them to what you expect. And oftentimes this works really well. I had a colleague who had a baby born preterm and quite low birth weight, and she was actually a developmental psychologist, so she knew the risks. She was very aware of both the risks and the kinds of treatments that were available. And within a year and a half, there were no negative outcomes after that point. And so that's not necessarily always going to be the case, depending on the
kind of care and treatment that is available, depending on how low the weight is or how early the birth is as well. But there are a lot of ways. Humans are pretty resilient. It's great if we don't have to rely on that resilience, but humans can be quite resilient. All right, so jumping back to the parents of the child, the biggest complaint after birth is the lack of sleep. This was definitely my biggest complaint, even with children who are quite a bit older. Lack of sleep is probably still my biggest complaint, and that they won't always eat the things that I make for them. I would really like them to eat more healthy foods, but that's why they've got gummy vitamins, right? Anyway, so lack of sleep is a primary complaint of people who have just given birth. So that can increase stress overall. So when you have less sleep, your body has higher stress hormones. Overall, you've probably seen this in your own life. If you do a couple night overnighters or like all-nighters before studying for a test, you've got impaired decision making. You might have depressive symptoms. If you have this long enough, it can even lead to depressive episodes. You fight with people, so you might have marital conflict. And so there are a lot of emotional and psychological adjustments after giving birth. So for the person giving birth, in the moments and couple weeks after birth, there are dramatic hormone shifts. So throughout the pregnancy, hormones have been shifting in the person who has the embryo and has the fetus. So have been even pretty quickly shifting over that time. Then right after birth, there's a sudden decrease in a lot of the hormones that went into forming that fetus. So they're over the first couple days, over the first couple weeks, dramatic shifts in hormones. And this can lead to challenges in psychological adjustment. So 70% of people experience what is defined as postpartum blues. This is basically symptoms of anxiety and depression, but that only lasts a short time, a couple weeks. So, and this usually appears 2 to 3 days after birth and can last for a couple months. Usually, for most people, these symptoms subside in about 2 weeks. Again, there is kind of a range there. And for a lot of people, about 10% of people who give birth, they experience postpartum depression. So those postpartum blues don't go away, even get worse, and it would be defined as depression, a major depressive episode. So for anyone giving birth, this is something to be aware of as a person who might get pregnant or as a person who might be sibling or friend or partner to someone who is pregnant. Most people who give birth, 70%, experience this postpartum blues. So be aware, and this might even be over a month. It usually subsides after a couple weeks, but if it doesn't, if it would be defined as postpartum depression, at that point, you definitely do need treatment from a psychologist. So I don't know why this is cutting off so many slides today. I double checked this a few hours ago. Totally fine. Yep, psychologist or psychiatrist. That's what I'm saying. So with that being said, do be aware, be responsive, because remember, these first couple weeks, these first couple months, you're adjusting to a new being in your life, adjusting work schedules, adjusting sleep schedules. You might really have no sleep schedule. You're just sleeping when you can. And that, in addition to these depressive symptoms, can be very, very devastating. So be aware that this is a very common challenge that people who are pregnant face, and to be there to support them in the way that you can. In some cases, especially if this has gone on for more than a couple weeks, definitely encouraging them to get treatment from a psychologist, a psychiatrist. Definitely, at the very minimum, talk to their prenatal care doctor at the very minimum after this. So with this, we do see this in media. So this is a couple comic panels from Daredevil. So if you know the comics behind Daredevil, Daredevil's mom did have postpartum depression and left, basically abandoned the family, went off, depending on the story. Some cases it's different, right? It's comics, there's a whole bunch of variations. But this is a couple panels where the mother is now apologizing to her adult son and talking about how he realizes that
postpartum depression is a real serious depressive episode, that it's not just some simple thing. It's not just that you abandoned us because you're a terrible person or that's what you chose to do, that you were not surviving, you were not thriving during this postpartum period. So be aware of that. I'm glad some media is showing that and people are more likely to talk about it now. A lot of aspects of our psychological health are being, it's more socially acceptable to discuss them more openly, which I think could be really good and increase people's treatment. And as we get to talking about the stigma with mental illness, as we go through the class, we'll talk about that more. So with this, the partner who didn't give birth may also experience postpartum depression. It's something that you might not realize. It's not as high of a rate, as common as it is for the person who gave birth, but their hormones actually change too. So the partner has differences of hormones as well during this pregnancy process and after the birth process. Their hormones change as well, not as dramatically, but they are at risk for postpartum depression as well. So, seeing a therapist, whether it's a psychologist, a counselor, social worker, a psychiatrist, there are a lot of really effective treatments for this. So be aware and be aware of this for friends, family members, yourselves, your own families, that this is a common thing to intervene for. And so that is the information that I wanted to share for Chapter 2. We've gone through a lot of both genetics and brain development in prenatal development and a few bits of the environment as well. So hopefully you're starting to see some patterns as we're going through these topics in lifespan development of how the environment as well as the individual all interact and can lead to lots of potential positive as well as negative changes over time.